Good morning everyone. Today I am going to discuss about platelet, its formation, structure, composition and function. This topic is included in the syllabus of major course of second semester of Bankura University. Platelets are blood corpuscles. We already know about the blood cells. There are three types of blood cells: RBC, WBC and platelets. Among them, platelets are involved or associated with the blood clotting mechanism. After cut or injury, when blood capillaries are damaged or blood capillaries are injured, blood comes out from the capillary through the injured portion. To minimize the bleeding or clotting, platelets are involved. This overall mechanism of blood clotting is known as hemostasis. Suppose this is a blood vessel. Around the blood vessel there is a layer of nucleated cells. These are known as endothelial cells. When the blood vessel is ruptured or damaged, the endothelial cells are displaced. Through this portion, blood cell wants to come out. To minimize the bleeding, platelet acts three ways. One of them is vasoconstriction. Secondly, platelet aggregation. And finally, coagulation of blood. Coagulation is the final process to seal the cut end. First of all, we have to know the identity of platelet. How platelet is formed in our blood. Platelet is formed in our blood from red bone marrow. Our red bone marrow contains stem cells. These stem cells are known as uncommitted pluripotent hemopoietic stem cells. This uncommitted pluripotent hemopoietic stem cell. Hemopoietic means because these cells are designated for the production of blood cell. Hence, these cells are known as hemopoietic stem cells. These uncommitted pluripotent hemopoietic stem cells starts to proliferate by mitosis cell division and produces some committed pluripotent hemopoietic stem cells. These committed pluripotent hemopoietic stem cells are of two types: lymphoblast cells and colony-forming cytoblasts. Lymphoblast cells are associated with the formation of lymphocytes. There are two types of lymphocytes, T lymphocyte and B lymphocyte, which are associated with our acquired immunity. T lymphocytes are associated with cell-mediated immunity and B lymphocytes are associated with humoral immunity. Right? After infection, T lymphocyte produces some toxic materials and produce a cytotoxicity to destroy the already infected cell to minimize the spread of infection. On the other hand, B lymphocytes are responsible for the production of antibodies and memory cell against the antigen. On the other hand, colony-forming cytoblasts produces some colony. This is called colony-forming blastocytes or colony-forming cytoblast because this cell can produce different types of colony. First colony is colony-forming unit E. E for erythrocyte. That means the cells of colony-forming unit E are responsible for the production of RBC. Colony-forming unit GM, G for granulocyte and M for monocyte. The cells of colony-forming unit GM are responsible for the production of granulocytes and monocyte. What are the granulocytes? Neutrophil, eosinophil and basophil. And the last one is very much important for today's topic, colony-forming unit M. M for megakaryocyte. The cells of colony-forming unit M are associated with the formation of megakaryocytes, and the megakaryocytes can produce platelet. Megakaryocytes are large shaped cells, large cells. Suppose this is a megakaryocyte. Due to the large volume, it can produce so many pseudopods and so many invaginations throughout the cell. That means this cell will be there are so many invaginations throughout the cell. Depending upon the invagination, the cytoplasmic fragments are separated from the cell, and these separated cytoplasmic membrane bound fragments are known as platelets. That means platelets are not a cell. Platelets are the part of cytoplasmic fragment of cell. Clear? Cytoplasmic fragment of megakaryocyte. A single megakaryocyte can produces up to 4,000 platelet. A single megakaryocyte can produces up to 4,000 platelets. Understand? So we have to know about the structure of platelet. Now we have to know about the structure and composition of platelet. This is a single platelet. Platelet is not a complete cell because it does not have any nucleus, but it is also membrane bound structure like a cell. So the composition of cell membrane of the platelet is same as the cell membrane, but we can tell the membrane which surrounds the platelet the surface membrane, because platelet is not completely a cell. It has cytoplasm like other cell, different cell organelles like other cell, and it also contains some protein factors inside the cytoplasm, and these protein factors are two types: alpha granules and dense granules. Alpha granules contain VWF, PDGF, and VWF, PDGF, fibrinogen, etc. On the other hand, dense granule contains thromboxane A2 and ADP. There are so many functions of alpha and dense granule, we will discuss later. There are some invagination throughout the platelet, and these invaginations are associated with the secretion of alpha and dense granule. Platelet also has a cytoskeleton. This cytoskeleton is composed of tubulin protein that provide the spherical or the disc-shaped structure of the platelet. These are the three major functions of platelet, which I already have told: vasoconstriction, platelet aggregation, and coagulation. When there is an injury on the capillary or vein, collagens of endothelial cell are exposed. This collagen binds with von Willebrand factor or VWF, and that will again bind with the VWF receptor, which is present at the surface membrane of the platelet. Then platelets are activated, and the activated platelets secretes some hormones, that is 5-HT, serotonin, adrenaline, etc. And all these hormones are vasoconstrictor. That means just after the cut or just after the injury, to minimize the bleeding, activated platelet secretes serotonin, secretes adrenaline, secretes 5-HT. That will result the vasoconstriction to minimize the bleeding. Secondly, the activated platelet secretes some dense granules. Dense granules contains ADP and thromboxane A2. This ADP and thromboxane A2, there are some receptors at the surface of platelet against the ADP and thromboxane A2. This ADP and thromboxane A2 binds with the receptors of another platelet to activate the another platelet, and togetherly these are known as PAF. PAF means platelet activating factor, because these molecules activate another platelet. All the activated platelet increase their diameter and produces so many invaginations, and they aggregates with one another. There is a specific mechanism of platelet aggregation, I will discuss later. Platelet aggregation. This aggregation of platelet produces a platelet plug. This platelet plug resists the bleeding temporarily. The third and the final function of platelet is the activated platelet triggers the formation of prothrombin activator. Triggers the formation of prothrombin activator. This prothrombin activator converts prothrombin of our blood into thrombin, and thrombin is associated with the conversion of fibrinogen into fibrin. Firstly, the fibrin produces fibrin monomers which are soluble, and then they are converted into fibrin polymer by polymerization, and the fibrin polymer is insoluble protein fragments. Understand? And finally, blood is coagulated and form the blood clot that will stop the bleeding. Platelet also secretes a component which is known as PDGF, or platelet derived growth factor, that is a potent mitogen that will trigger the mitosis cell division around the wound. As a result, these cells starts to proliferate them very rapidly by mitosis cell division for wound, to trigger the wound healing process. Now see here, after a cut or after an injury, this portion will secrete some collagens, will release collagen. Collagens are exposed, and with the exposed collagen, now the damaged endothelial cell secretes VWF. VWF means von Willebrand factor. The VWF binds with the collagen. VWF binds with the collagen. Suppose this is VWF. Each platelet surface contain VWF receptor, that is known as GP1V receptor. Each of the platelets are here. These are inactive platelet. Each inactive platelet express GP1V receptor on their surface. The inactive platelet then binds with the VWF with its GP1V receptor. This is GP1V and this is VWF. As a result, after the binding of VWF with GP1V receptor, the inactivated platelet become activated. What is the difference between inactivated platelet and activated platelet? After activation, platelet increase its diameter and its volume and produces so many invaginations throughout its surface. And through this invagination, platelet release two types of granules, that is alpha granule which contain, alpha granule contains number 1 VWF, number 2 fibrinogen, and number 3 PDGF. There are some receptors present on the surface of another platelet. The receptor is designed against VWF is known as GP1V. The receptors are associated with the binding with fibrinogen are known as GP2V or GP3A. All these receptors are present on the surface of activated platelet. PDGF is a potent mitogen that will trigger the mitosis cell division of the endothelial cells of blood vessel. to proliferate very rapidly and accelerate the wound healing process. On the other hand, the activated platelet also secrete dense granules. Dense granules. There are two types of dense granules. Number one is ADP or adenosine diphosphate. Another is thromboxane A2 or TXA2. There are also some receptors against ADP and thromboxane A2. The receptor is specially designed against ADP is known as P2Y12 receptor, which is present P2Y12 receptor which is present on the surface of another platelet. Thromboxane A2 will bind with TP receptor. TP receptor. This is also found on another platelet. When a single platelet is activated, that will secrete both the alpha and dense granules for the activation of another platelet. Understand? When a single platelet is activated, that will release these granules for the activation of another platelet. On the surface of another platelet, suppose this is TP receptor, suppose this is P2Y12 receptor, P2Y12 receptor. When TXA2 or ADP binds with TP or thromboxane A2 receptor, adenyl cyclase activity of the another platelet will increase and produce a cyclic AMP that will activate this platelet, and this platelet is also activated. Activated platelet also exposed to receptors on their surface. These receptors are known as GP2B and GP3A, and these GP2B and GP3A receptors are associated with the binding of fibrinogen. Suppose this is GP2B receptor, GP2B, and suppose this is also another GP2B or GP3A or GP2B receptors. Both the receptors are associated with the binding with fibrinogen. Fibrinogen binds with both the receptors to connect these two platelets. By the way, all activated platelets are attached with one another, and fibrinogen act as the linker molecule. Understand? GP2B, GP3A, both receptors are the fibrinogen receptors present on the surface of activated platelet. When fibrinogen connects both the platelet by binding with GP2B or GP3A receptor, the activated platelets are attached with one another. This is known as platelet aggregation. Cytoplasm of platelet also contain actin and myosin. This actin and myosin compress this aggregation to produce a compact aggregated structure that will temporarily seal the damaged portion to prevent bleeding or to minimize the bleeding. Understand? And the last function of platelet is coagulation of blood, and this is the final. Coagulation of blood can be discussed by three stages: stage 1, stage 2 and stage 3. Stage 1 is associated with the formation of prothrombin activator. Stage 2 explain the conversion of prothrombin into thrombin, and stage 3 explain the formation of fibrin clot formation. In the stage 1, the process of coagulation can be initiated by two ways. either intrinsic pathway or by extrinsic pathway. When the coagulation process or the stage 1 is intrinsic pathway, when the platelet or the components of our blood vessel initiate the process is known as intrinsic pathway, but when the damaged tissue initiate the process is known as extrinsic pathway. In case of intrinsic pathway, the damaged endothelial cell is responsible and the platelets is responsible to initiate the process of coagulation. Collagens are exposed from the damaged endothelial cells, and the exposed collagen activate factor 12 with the help of high molecular weight kinogen or HMW kinogen. Activated factor 12 also activate factor 11. This is known as this cascade mechanism because the activation of a factor activates the another factor. Hence, this is known as a cascade mechanism. The activated factor 12 activate factor 11, and the activated factor 11 will activate factor 9 into activated factor 9 with the help of calcium ion. Then activated factor 9 will activate factor 10 with the help of calcium ion and factor 8. This activated factor 10 produces prothrombin activator with the help of factor 8 and phospholipid, which is secreted from the activated platelet and produces prothrombin activator. On the other hand, in case of extrinsic pathway, the damaged tissue initiates the process of blood coagulation. From the tissue trauma or the damaged tissue will secrete glycoproteins and phospholipids that will activate factor 10 with the help of factor 7. Same as the activated factor 10 produces prothrombin activator with the help of factor 5 and calcium ion. This prothrombin activator converts prothrombin into thrombin in the second stage, and thrombin provides a positive feedback to form more amount of prothrombin activator. In the third stage, thrombin converts fibrinogen into fibrin monomer. Then polymerization of fibrin monomer produces a fibrin polymer that will prevent the bleeding and form the blood clot. This is final. I have discussed the platelet, the function of platelet, the structure of platelet, the composition of platelet, platelet aggregation or activation of platelet, and coagulation process. I hope all of you understand. Thank you.